We are preparing for the SeekOne DD single-cell multiplex ligation-based probe sequencing experiment. With rehydration complete, we now proceed to nuclei isolation. After removing the supernatant from the final rehydration step, add 1 milliliter of NLB buffer to the glass homogenizer tube. Transfer the tissue into the homogenizer tube and ensure the sample is fully immersed in the buffer. The sample is now ready for mechanical homogenization. Begin a 5-minute incubation on ice at the start of homogenization. Homogenize the tissue using pestle A with steady vertical strokes, followed by pestle B to further dissociate the sample. Continue homogenization until the suspension appears uniformly opaque with no obvious tissue fragments remaining. For difficult or highly fibrotic tissues, additional strokes may be required. Once homogenization is complete, place the sample back on ice. Add 1 milliliter of WB1. Mix gently. Pre-wet the sterile strainer with WB1 before filtration. Filter the suspension to remove debris and obtain a single nucleus suspension. Rinse the cell strainer with 2 milliliters of WB1 to maximize recovery. Discard the used strainer and cap the tube. Centrifuge the filtrate at low temperature to Pella Nuclei. Carefully remove the supernatant while keeping the tube on ice. Add 1 milliliter of WB1 and mix thoroughly to ensure a homogeneous nuclei suspension. Carefully pipette 5 microliters for nuclei counting. Keep the remaining nuclei suspension on ice for 5 minutes during counting. Load into the fluorescent cell counter for automated nuclei quantification. Use the counting results to assess nuclei yield. For optimal downstream processing, a yield greater than 500,000 nuclei is recommended. Centrifuge the sample at 4 degrees Celsius for 5 minutes to pellet the nuclei. For samples with yields below 200,000 nuclei, increase centrifugation to 2000 G. Carefully remove the supernatant and resuspend the nuclei pellet in WB2. Mix thoroughly and perform counting. After nuclei counting, proceed according to your experimental workflow. For immediate processing, continue directly to pre-decross linking. If the sample will not be processed within 2 hours, pellet the nuclei again, resuspend in buffer Q, and store at -80 degrees Celsius for up to 30 days.